Thank you.
Thank you. So I'm going to be talking about research ethics in terms of genetics and genomics, but like the last speaker and a couple of others before, it's absolutely impossible to split diagnostic ethics from research ethics in this field. So I'll really be talking about both. And this is what I'm going to mention, a special peculiarity about genetic testing. I'm going to touch on incidental and secondary findings and the definition thereof, a little bit about non-disclosure and direct-to-consumer testing. So if you think about, have I got news for you, and they have, who is the odd one out, and if I asked you to look at these 4 photos and thought which one looks the most different, you probably would pick the MRI scan, but as a geneticist, I would say bottom left, genetic testing is also the odd one out. Why? Well, if you think about other testing, the other blood tests, your thyroid function tests, your hemoglobin tests, and even your MRI, they change over time. It goes up and down. You're forever having your bloods done. It changes with treatment. Tests are repeated constantly. Your brother won't mind you having your LFTs, your MRI done. The interpretation of your blood or your MRI does not require sampling family members, and your results are freely available on the hospital systems. On the other hand, genetic test is really one test for life. You better get it right. You can go back to the data and reinterpret it, but it's really one test for life. And you have to treat it like a blood transfusion, with the identifiers are very, very strict because of that. The consent is required as part of the Disability Act. It's the highest risk registry on the GDPR, so we spend our time tripping over GDPR issues at work. And interpretation may require DNA samples from your relatives, parents, siblings, etc., which might be difficult to get because they might be deceased, or it might have been an ART pregnancy, or you might have no contact with the father. And there are implications for your family members. Your brother might mind you going off and doing something and finding something that implicates him or her, your sister's health too. The results will never go away. Sometimes the interpretation might change. Whereas I don't believe the interpretation of a thyroid function test is going to change. And when you're doing testing of parents, it might reveal non-paternity. Did you mention that when you took the bloods from the parents? Because sometimes we trip over ourselves with regards to that. And believe you me, the results are not freely available in hospital systems, which also causes us problems. So just some dangerous assumptions. Anybody can order genetic testing and consent is complete or informed. The other one is the parents are the biological parents. The other one is you're going to get a very black and white result. Well, sometimes it can be very much shades of grey. And the wording on the report is really, really important, because sometimes people do not understand what is written, the difference between the scientist and the clinician. The other thing is to assume the abnormality you find explains the phenotype in your patient. It mightn't. It might be a benign finding, and it can be difficult to work that out. And then the other one people make the mistake is they get a normal result, and they assume it means no genetic diagnosis. It doesn't mean that. It just means you haven't found it sometimes. And then, of course, a research result needs validation. It's not the same as a clinical diagnostic result. And then the final one is direct-to-consumer tests are accurate. They're not. So just to kind of give you a little bit of background to human beings, so we used to do tests called chromosome tests years ago, and we still do them. They're still required. Nothing has superseded them in certain circumstances. And in some ways they were great. They gave us a diagnosis and they gave us very few false positives. But as the testing has got more and more sensitive, and we're down to picking up literally variants in ACs, Gs and Ts with full sequencing, the number of false positives has gone way up, but of course the diagnostic rate as well has increased. And the reason the false positives have gone up is because all of us are full of variation. So each of us have 23,000 genes. That's about 2 to 3 billion base pairs. Each of us have 2 to 3 million variations in our DNA compared to the normal human being. And the difficulty sometimes for laboratories and clinicians is to determine whether a variation that's flagged, whether it is disease-causing or whether it is benign. And the other interesting thing is every newborn baby has around 50 new DNA changes that the parents didn't have. And that's just part of evolution. The other thing just to mention is we no longer use the word mutation. We're not meant to. We use variant. So this is the ACC e-wheel, which is really for use for R&D moving into diagnostic labs, but it's a great simplifying factor in terms of when a lab starts offering tests and what an R&D would involve. So the first thing is the A stands for analytic validity is the test you're doing, is it actually going to pick up the variant you think you're meant to be picking up? You know, you have to have something that's definitely going to work. And then the second thing is, what you're picking up, is it really associated with disease? Do you know that, and is that well quantified? There's no point picking up a variation if it doesn't mean anything to the patient. The third one is, what you're picking up, is it worth offering as a diagnostic test? Because, like, for example, there's no point offering eye colour gene tests. That's not going to affect the patients. It has no clinical utility. And then, of course, the E stands for ethical, legal, and social issues. So on the left there is a publication from the European Society of Human Genetics about guidelines for diagnostic next-generation sequencing. So you can imagine moving from research next-generation sequencing. It was even less robust for this. And essentially, they state that the platforms aren't yet stable, which means you're going to get some errors. You have to be well aware of that. But it's still worth bringing into the diagnostic setting. But just be well aware that there are sort of risks when you're bringing in new technology. The other thing is with genetic tests, you're meant to pre- and post-test counselling. That's mentioned in this paper and does talk about lab frameworks. So just incidental and secondary findings. The most important thing is to understand there's a difference between them. So the European Society of Human Genetics announced their secondary finding policy in 2020. And there's a big difference between the American College of Medical Genetics screening and European Society of Human Genetics in terms of secondary findings. So incidental findings are something that you can't help but find, and we have to deal with them, and that's okay. So it's like you did something like a chromosome array test, which looked for bits missing or bits extra of the genome, and you found the cause of the problems that you did the test for in the first place. We would tend to do it on children with intellectual disability, and you might find a huge big deletion, and within the deletion happened to be a cancer gene. Well, you have found the diagnosis in your patient, but coincidentally, you've happened to have found a finding that's relevant to that child's health. And they are accepted as a consequence to testing, but, and there's no argument between Europe and the States on that. A secondary finding, which is also called opportunistic genomic screening, is where you do a purposeful search for other health findings, variants in the DNA that are completely unrelated to the patient's phenotype. Now, secondary findings are illegal in France. There's different legalities in different countries in Europe, but in the States, the laboratories are obliged to publish them unless the patient opts out. The European Society of Human Genetics have suggested that we should have in Europe an opt-in policy. And they say particularly if you're doing a test on a child, on a minor, you should really, really consider whether you want to find secondary findings or not. And I have to say, as clinical geneticists in Crumlin, we never tick the secondary findings box because we have to deal with it, and we don't understand the consequences of that. And if the specialist, which is us, does not understand how to inform our patients on really what it means, there is a problem in there. So you might think picking up pathogenic variants opportunistically, is that not a good thing? What's the big deal? Aren't you preventing disease? Isn't it good to identify disease variants and prevent disease happenings? And am I being paternalistic? Well, maybe I am, but I'm the specialist who's going to advise you, and I don't know how to advise you. And that is an issue. So, you know, there's a lot of papers here I'm citing from Caroline Wright. She works in Exeter, and she's done some great work using UK Biobank, which is nearly 500,000 samples of healthy individuals. And one of the things that she's noticing is that the penetrance of these variants in a population cohort are very different to the patient cohort where there's a family history. So we don't really know what risk to give somebody and they mightn't have a risk at all, who's in inverted commas, pathogenic variant. And on the second paper that she's also co-author on, the interpretation, they very much advise that the family history is key in interpreting whether or not somebody is likely to develop a disease. So the family history, which is the cheapest test you can do, is still a really fundamental part of interpreting these variants. So our secondary findings happening in Ireland. Yes, we did a 6-week audit last year, and 4 people were referred in for us to manage them, to pick them up. And what's the consequence to this family? So we really feel quite uncomfortable because we don't really know the penetrance without a family history, and we don't know really what to say to the family. And then there is a risk to that person's relatives of issues. This is a really interesting paper. It's not yet fully published, but it's again by Caroline Wright, and she's showing that in 2 genes, and there's 23,000 genes, so there's a lot of work to do, but in 2 genes that predictive loss of function variants shouldn't be described as pathogenic because they found people had an alteration and that it was causing what's called, different splice site alterations. So it actually wasn't pathogenic at all. So in other words, you'll find one variation is pathogenic, and then you have to go searching to see in the same gene whether there's something protecting that patient. And we're going to have to do that for 23,000. They're the two genes that they had done it in, and it's just about to be published. So in the absence of knowing what to say to these families, maybe don't do the test. So moving on now to non-disclosure. Non-disclosure is where somebody has known to have a hereditary disorder. It has consequences, potentially has consequences to their siblings, to their children, and will they reveal the diagnosis to the family members? Now, you could argue if somebody's in a research project and got a research result, if it wasn't validated, maybe they aren't obliged to tell their relatives, but we still would obviously like them to. In the diagnostic setting, we come across this occasionally. A lot of times it's parents trying to protect their children. I can tell you most of the incidents where we've found this, the child already knows. So the parents are trying to get their children tested for the genetic condition that might predispose them to cancer or something, and doesn't want to tell the children that, but actually a lot of times the child will turn around to the parent and say, look, I knew this all along. Even small kids of 8 and 9. The other ones are things like, siblings, cousins. I mean, that's very difficult to be contacting your relatives about something that might be very private to you. The illness in your family is very, it's private information, and it is difficult to be telling relatives that, you might be at risk of a chromosomal translocation or whatever. We do to whom it concerns letters to try and facilitate, to help that family, say, to pass it on to the relatives, but we spend our time tripping over GDPR, because ideally we would like to name the person who's got the condition, so that if that sample from the relative comes in, we know exactly what we're testing for, but then that breaks privacy too. So it is a very difficult area. And just to show you, we published this paper, it's quite a while ago now, looking at relatives of people with BRCA1 and BRCA2 mutations to see what percentage turn up, and it's actually very small. I think it was just about 30% of females, and there, if I'm reading there, 19% of male siblings of the whole cohort. They were the only ones who came forward for testing. And what we noted was that daughters of mothers with BRCA1 and 2 were much more likely to come forward than testing than siblings of mothers than were male siblings of mothers. Not surprisingly, I guess that's the protective element. But it just shows even within a nuclear family, something like BRCA, not everybody comes forward for testing. And direct-to-consumer testing, so we have Ancestry.com literally across the Liffey. I think it's their European headquarters. And so these are the things that you might be considering buying for Christmas. Maybe reconsider. So they sell it as an informative test. This is a great paper, actually, in the British Medical Journal a number of years ago. So they sell it as, this will inform you about your health. So the one thing is that they're full of false positives. They're full of false negatives. It depending on the testing methodology, and this testing methodology is sometimes used by research groups too, which is why it's so important to validate a research test. So a lot of them use this SNP technology to inform you that, you've got a risk of this or that or the other. And I can tell you the only geneticists who ever use direct-to-consumer testing do it for fun, and then they give us a great hilarious presentation at a genetics conference and we all laugh at it. We would never believe anything from these results. So this, back to the wonderful Caroline Wright again and her group, looked at SNP chips, which is what these DTC companies use, and some research groups. I don't mind the research groups doing it. They're doing it as a first baseline. But it should never, ever be used. Her bottom line, they're extremely unreliable for genotyping very rare pathogenic variations and should never be used to guide health implications. So that's all very well, but we get referrals now for people with DTC. So one of my colleagues had one where this wife bought her husband a birthday present of a DTC, and he went and tested himself, and he found supposedly he was a carrier of an X-linked condition that caused neurological problems later on in life, and it absolutely panicked him and his wife. Got dumped in on us to sort out. We repeated the test. It was completely erroneous. He wasn't a carrier of anything. Took up a huge amount of our time. It cost HSC, et cetera, et cetera. So the problem for us ethically is, do we take up these valuable slots ahead of families with a genuine high risk? Now, I do have sympathy for the couple, but we do have that ethical dilemma. We're a publicly funded service. We have long waiting lists. And the other issue I guess I have is this was a false positive, but we're not going to be referred to false negatives. So one of that paper from the BMJ talks very clearly about if you're a GP and you have somebody with a strong family history still refer even if their DTC test is negative because it was probably wrong. And we're never going to see the false negatives unless the GP is very astute about that. And of course, GPs are not equipped. It's not their job either to fix these DTC issues and to arrange the retesting. So there's a big ethical issue there of what we do with them. And that's it.